Hello students, I am Dinesh Udupi. This is session 20 of Molecular Biology, Evolution and Ethology paper. In this session, we are going to study about B and T lymphocytes and the immune response, especially the antigen-antibody reactions. Let us start with the understanding of B lymphocytes. We already know what are B lymphocytes. In the previous sessions where we studied the humoral mediated immune response and cell mediated immune response, we came across these words B lymphocytes. We studied about these cells which are involved in humoral type of immune response. They are a type of white blood cells which are involved in the humoral mediated immunity. They get converted into plasma cells and secrete antibodies involved in the immune response. We already discussed in detail how the humoral mediated immune response occurs. Whenever the pathogen enters into the body, there is a mechanism by which the B lymphocytes they get selected, they get activated, and these B lymphocytes they get converted into plasma cells, and these plasma cells start secreting antibodies. Now we can say B lymphocytes are the ones which are involved in the production of antibodies against a particular antibody. I am very sorry. They are involved in the secretion of antibodies against a specific antigen or a specific pathogen. They develop and mature in bone marrow. B lymphocytes are primarily produced in the bone marrow, and once they get sensitized or mature, they leave the bone marrow and they enter into the lymph or blood circulation. The human body makes millions of different types of B cells each day that circulate in the blood and lymph, performing the role of immune surveillance. Now, in the previous session, we studied about one concept of clonal selection theory. What happens is millions of different kinds of B lymphocytes are produced in the body every day, and they are waiting for the antigen, and they are waiting for the stimulation and maturation. And some of the cells they circulate in the blood and they do the surveillance, and they are always circulating and they are searching for the antigen or the pathogen. They do not produce antibodies until they become fully activated. Mature B cells differentiate into either plasma B cells or memory B cells. This one we already studied in the previous session. Once the B cell get activated by a particular antigen, then it is going to mature into a plasma B cell or a memory B cell. You know the difference between plasma B cell and memory B cell. Plasma B cell starts producing antibodies. So plasma B cells are involved in the production of antibodies, whereas memory B cells, they remain in the circulation for a long time. They remember that specific antigen. These memory B cells are very important as far as immunity is concerned, because they are going to respond when the person gets infected by the same pathogen for the second time. Now, we already studied about the secondary immune response. So, when the person is infected for the second time, the memory B cells will identify the antigen very quickly and the immune response will be very fast, and the person normally will not show any symptoms and he will not suffer from the disease. That is the importance of memory B cells. These memory B cells may remain in the blood circulation for few hours, few days, few weeks, few months, or there are some memory cells of particular pathogens which will remain in the blood circulation for many years. That means once the person is infected by a particular disease for the first time, he remains immune to that particular disease because of the memory cells for a long time. The B cell may either become one of these cell types directly, or it may undergo an intermediate differentiation step. So the conversion of the B cell into plasma cell or memory B cells may be a direct process, or it may involve some intermediate steps. That is about the B lymphocytes. Now let us move on to the study of T lymphocytes. This is also a type of white blood cell. It is mainly involved in killing pathogens directly, whereas the B lymphocyte is involved in killing the pathogen through the mediation of antibodies. Here, T lymphocytes will directly attack the pathogen and they will destroy it. They are formed in bone marrow like B cells. T lymphocytes are also formed in the bone marrow, but they migrate to the thymus and differentiate. Since they migrate to the thymus and the differentiation occurs in the thymus, they are called T lymphocytes. That letter T is because of the maturation organ, that is thymus. They are involved in cell-mediated immune response. Now remember, B cells are associated with humoral mediated immune response and T cells are associated with cell-mediated immune response. Each T lymphocyte will develop its own T cell receptor that is specific for a particular antigen. Now, during the cell-mediated immune response process, the T lymphocyte, each T lymphocyte will get sensitized and get specialized, and it will develop its own T cell receptor that is specific for that particular antigen. Once activated, the T cell will proliferate and differentiate into an effector T cell. Effector T cell is the one which is going to do some action, going to destroy the pathogen. The sensitized and matured T lymphocytes will modify themselves into the following types of T lymphocytes. They are cytotoxic T lymphocytes, TC. They kill target cells directly. So cytotoxic T cells or cytotoxic T lymphocytes are the ones which are capable of killing the target cells directly. Then T helper lymphocyte. We also call them as helper T cells. Now we understood the role of helper T cells in the immune response. They are involved both in humoral mediated immune response and cell mediated immune response. They identify infected cells. They act like messengers. So they will identify the antigen and they will pass on the message to the B cell and T cell so that the B and T cells will further modify and show the humoral mediated immunity or cell mediated immunity. Memory T lymphocyte, as we already discussed, they will remain in the blood for a long time, and once the person gets infected, in the first time he will develop the memory T lymphocytes, and in the second infection the memory T lymphocytes will see that the reaction, the immune response is very quick and the person doesn't suffer from the disease or doesn't develop any symptoms. In the humoral mediated immune response, antibodies are produced by the plasma cells. These antibodies are going to react with antigen, and that reaction or those reactions are called antigen-antibody reactions. In the humoral mediated immunity, antibodies are produced. They interact with the antigens and neutralize the antigens or the cells that carry them. Following are the types of antigen-antibody reactions. So these the antigen-antibody reactions can happen in two ways. One is precipitation and other one is agglutination. So precipitation and agglutination are the two main types of antigen-antibody reactions which are going to neutralize the pathogen. First one is precipitation. When an antibody binds to a soluble antigen, the antigen becomes insoluble and it may precipitate or float in the fluids. If antigen-antibody complex precipitates, it is referred as precipitation reaction. What is happening here? The antibody binds with antigen molecules. The binding of antigen and antibody molecules will neutralize the effect of antigen. Suppose the antigen is a toxic molecule. If it is not bound with any antibody, it may cause some damage. It will show its toxic effects. When it gets bound with the antibodies, it is neutralized. Its effect is gone. So it becomes harmless. So that is how the precipitation reaction is going to protect the body against the toxic materials or the antigens. The second type of antigen-antibody reaction is agglutination. Aggregation of cells due to antibody binding is known as agglutination. Here what happens? The pathogens, you think of bacteria. Now bacteria carry antigens on their surface. Those antigens are identified by the antibodies. The antibodies will come and bind with the antigens which are present on the surface of bacteria, and then when all the bacteria bacteria surface are occupied by the antibodies, all the bacteria will clump together. When the bacteria are clumped together, that condition can be considered as agglutination. Here also, bacteria loses its pathogenicity. The bacteria becomes neutralized. Once the bacteria are neutralized by the agglutination, then they can be destroyed by the further processes. Here, in this picture, you can see this is agglutination. You can consider these as the bacteria and these are the antibodies. Here, many bacteria and the surface bound antibodies together have made the bacteria immobile. All these bacteria are immobile and now they will not show their damaging effects. Here, this is precipitation. These are the antibodies. Y-shaped molecules are antibodies, and these are antigens bound with each other. So, binding of antibodies to the antigen molecules has created one soluble, insoluble precipitate. So, this is called precipitation. Here, the antigens are neutralized, and here, the pathogens are neutralized. Through this chart, let us just try to understand the importance of agglutination and precipitation reactions. Both agglutination and precipitations are examples of antigen-antibody reactions, AgAb reactions. That means the formation of AgAb complex. We already understood the difference between precipitation and agglutination. So formation of precipitation and agglutination is the secondary stage. What happens after the formation of agglutination or clump or the precipitation? The agglutination will lead to opsonization. What is opsonization? We'll see. And precipitation will lead to lysis of the antigens. With the help of this picture, let us try to understand the difference between opsonization and lysis. Here, this is the picture where you can see the antibodies. This is bacteria. The surface antigens are identified by the antibodies, and the antibodies will bind to the antigen. That means the antibodies are going to bind to the surface of bacteria. When large number of antibodies are bound to large number of bacteria, it will lead to agglutination or clumping. These antibody bound bacteria are identified by immunologically active cells, the cells which are capable of destroying the pathogen. But the pathogen destroying cells, I mean the immunologically active cells, cannot go on destroying all the cells because they may damage the healthy cells of the body. So therefore, there must be a mechanism to identify only the infected cells or only the pathogen, and then that has to be destroyed. That is done with the help of complement fixation, and now these cells are going to identify the bacteria since it is coated with antibody. Since it is coated with antibody, it will be recognized as a pathogen or it will be recognized as a infected cell. Then these cells, the immunologically active cells, are going to engulf it, phagocytosize this. Once these bacteria are phagocytosized, they are going to be destroyed inside by the by the action of enzymes. So this process is called opsonization. The antigen-antibody reaction, which leads to agglutination, will end up with opsonization of the pathogens like bacteria, and bacteria are destroyed. This is how the immune system works in case of agglutination. In case of precipitation, you think of the toxin material, which itself, the molecule itself is treated as the antigen that is bound with the specific antibodies. And when large number of antigen antibodies clump together, it is going to form the precipitation. This precipitation is also identified by the immunologically active cells. But here the cells are going to be, the antigens are going to be broken down by the release of some enzymes. Now, when the enzymes are going to be released by the immunologically active cells, and the antigens become neutralized, they lose their ability to cause damage to the cells or the system or body. That is how the antigen-antibody reactions will lead to the protection of the body by means of opsonization and lysis. The antigen-antibody reactions in the body, they help in protecting the uh, tissues or body in general. But the technique, the knowledge of this antigen-antibody reaction is used in diagnosis process. For example, application of bacteria, for example, Lancefield grouping of streptococcus. The antigen-antibody reaction process is used in detection of the infection, whether the person is infected with a particular antigen, particular bacteria or not. You can identify using this uh, agglutination reaction or the precipitation reaction. Detection of antibody for diagnostic purposes, for example, VDRL in syphilis. Identification of syphilis, in the VDRL technique, they use the same process is used. The antigen antibody reaction, that concept is utilized here. Forensic applications in identification of human blood and seminal stains. The blood group of the person is identified, with the help of antiserum. What we do in blood grouping test, we use the antiserum A, antiserum B, and antiserum D. What happens? The clumping. If the blood clumps, then we identify the clumping and the blood groups. If antiserum A and blood clumps, then the person's blood group is said to be A, like this. The same principle is used here in the forensic application, in the general detection of blood group, or else in the forensic application where the human blood and seminal stains can be identified. Testing for food adulterations can be done to standardize toxins and antitoxins. To standardize the toxin and antitoxin thing, this antigen antibody phenomenon is used. The principle of antigen antibody reaction is used. That's about the antigen antibody reactions. In this session, we studied about B lymphocytes, T lymphocytes, and precipitation and agglutination as a result of antigen antibody reactions. Thank you.